Good morning. I'd like to thank the organizers for the invitation to speak today on the work that we've been doing on RNA splicing and ancestry-related molecular targets in precision oncology and cancer disparities. So let's talk for just a moment about cancer health disparities. These are the government definitions of health disparities, a particular type of health difference closely linked with social or economic disadvantage. The NCI's definition is below: adverse differences in cancer incidence, prevalence, cancer death, survivorship, burden of cancer, or related health conditions among specific populations. I've been working in this field for quite a few years, and I have my own definition: the unequal burden of cancer among racially and ethnically diverse minorities, the medically vulnerable, and underserved. We know now that this problem is local, regional, national, and global, and I think this talk is perhaps timely at this moment, given the national and international recognition of the magnitude and impact of health disparities, and in our field, cancer health disparities. So to give you a sense of what disparities look like, this is a focus on prostate cancer at incidence rates and death rates. There's good news and bad news in this slide. The good news is overall incidence and mortality rates are going down. But the bad news is that difference between the red line and all of the other lines illustrates the difference in incidence and mortality among African-American men compared to Caucasian and other racial and ethnic populations. Well, what does that look like in clinical or biological terms? It means earlier mean age at diagnosis, shorter mean survival, more common advanced stage diagnosis, greater risk of early biochemical recurrence, and more common metastases. What we've also learned is each tumor type and each subtype of each tumor exhibits its own nuances with respect to disparities in incidence, aggressiveness, outcomes, and mortality. To give you a sense of the magnitude of that disparity in prostate cancer, on the left is incidence rates, and you can see a 1.6-fold increased incidence rate among Blacks compared to whites. And on the right, you see mortality rates with a particularly startling statistic, a 5.1-fold difference in mortality rates between Blacks and Asian Pacific Islanders. So you can see with prostate cancer, this disparity is quite dramatic. Well, what are some of the contributors to cancer disparities? Some people use the words determinants of health or determinants of disparities. And we know that disparities are driven by a multi-level, multi-factorial group of contributors, including social and environmental aspects like personal health beliefs, socioeconomics, environmental exposures, lifestyle issues like smoking status, diet, exercise, obesity, comorbidities, and the physical environment. Structural issues, for example, our health systems, access to care, policy, both local, state, and federal, structural racism, and implicit bias within the health system. But we also know now that there is a biological contribution to cancer health disparities, which includes germline somatic differences in tumors, genomic, epigenomic, transcriptomic, proteomic, and metabolomic differences based on population. And today, I'm going to be focusing on the biological aspects of disparities, but I want to make sure that everyone is clear that these biological effects or aspects occur in conjunction with social, environmental, lifestyle, and structural determinants of cancer health disparities. And one of the most exciting developments in science right now is that this multi-level, these multi-level contributors to disparities illustrate the need for new methodologies in intersectionality and what's called convergence science, developing methodologies to understand how these various multi-level contributors impact each other, whether they mitigate or exacerbate cancer disparities. Well, when we talk about biology and race and cancer disparities, it's very important to quickly point out that race in and of itself is not a biological construct. Race and ethnicity are sociocultural constructs. However, racial ancestry, as a function of the human diaspora, whether the optional or forced in the context of slavery, affects cultural, phenotypic, genetic diversity, and therefore disease risk, biology, and outcomes. So again, in and of itself, it's not a biological construct. But as humans spread out around the planet, our phenotypes change. And those phenotypes were dictated by our genotypes. And the genotypes dictate our susceptibility to different diseases around the world, the biology of that disease, and differential outcomes as well. So to address these complexities, some years ago, I developed a vision for disparities impacting translational research that I call trenches, benches, bedsides, and wrenches, where the trench is the community, where we can actually learn from the community what the most important issues and problems are. We translate that information to the bench, and of course that can be either the wet lab or epidemiology or outcomes research. And then that information, as we learn, we want to translate to the bedside, which would improve treatment. But gradually we want to move the information back to the community. But in our model, we view it, we move it back to the community via policy, because we want to not only change the minutia of the disease, we want to change society so that we can impact all of those contributors to disparities. And so this is where we use the word wrench, just because it rhymes with trench, and we use it to refer to policy. Well, this is what our laboratory actually looks like. This is an arc that stretches from tissue and blood specimens collected from patients of different racial and ethnic groups, a focus on the biology, and today I'm focusing very specifically on RNA splicing. We utilize a number of different systems, siRNAs, CRISPR, small molecules, splice-switching oligonucleotides. We generate cell lines, xenografts, and PDXs from patients from different racial and ethnic groups, with the ultimate goal of conducting, and we have ongoing active race or ethnicity stratified clinical trials with associated correlative science. We have an umbrella biobanking protocol called GenCade, which stands for the Genomics of Cancer Disparities. We're also interested in education. We want to be training the next generation of disparities researchers. And here I'm just showing that I'm PI with Kevin Williams to a P20 Apache grant from the NCI, which links NCI-designated comprehensive cancer centers to historically Black universities and our partners at NCCU here in Durham. And these are just some pictures of our scholars. We have two projects. We're working on prostate and inflammatory breast cancer in African Americans. And we just recently were awarded a P20 SPORE on cancer disparities, with two projects, lung cancer and stomach cancer disparities. And this is just a snapshot of my lab. I'll be talking about the work from these postdocs shown here: April DeVoe, Muthana Al-Abo, Tyler Allen, Sean Pwarski, Joab Odura, Karen Wegerman. The lab is co-run by myself, Dan George on the left, but the person who really makes it happen is right in the middle. Dr. Jennifer Friedman is an astoundingly brilliant genomicist, and she runs the day-to-day operations and scientific directions of the lab. So I hope you can see that we not only talk about disparities and diversity and inclusion, but we walk the walk and have an exceedingly exciting and fun, diverse lab. So let's get back to splicing. How did we arrive at splicing? So we started this work early in the aught years, and first reported differentially methylated genes in prostate tissues from African American and Caucasian men in clinical cancer research, and then androgen receptor target genes in African American prostate cancer disparities published in Prostate Cancer, and then another paper in CCR on identification and functional validation of microRNA mRNA pairings in African American prostate cancer disparities. And that culminated in a Nature Communications paper in 2017 entitled Alternative Splicing Promotes Tumor Aggressiveness and Drug Resistance in African-American Prostate Cancer. So we're going to do a deep dive into splicing, and I want to start first with splicing 101, because I'm always intrigued to discover how many of my colleagues have forgotten the splicing lecture from Biology 101. So this is just a quick reminder that at the DNA level, our genes are discontinuous. They include exons and introns. During transcription, a pre-mRNA is the first RNA made. It includes both exons and introns. The introns have to be spliced out. This process was discovered in 1977, called alternative RNA splicing. And this is the process by which about 20,000 genes in the human genome produce about 250,000 proteins. And by splicing different exons together, you can produce multiple different proteins from the same gene, each with a different function. We know it drives evolutionary biological diversity, and therefore likely drives tumor-related biological diversity. And we know that the function of proteins produced from these variants can be dramatically different, as if they were made from different genes entirely. And just as an example, I'll just show you one example from the MCL gene. And if you look, there it comes up in the red box. And what you can see is just the removal or the alternative splicing of a single exon can convert that gene from a pro-apoptotic to an anti-apoptotic gene. And we now know that splicing impacts every single one of the major hallmarks of cancer. So splicing is very important, and it is regulated by a combination of cis-acting splicing elements at the DNA level, usually right at the junctions between the exons and introns, and a vast machinery, at last count nearly 500 different transacting splicing factors, which interact with the cis-acting splicing elements in the pre-mRNA, exactly duplicated from the DNA. Excuse me. And those two work together to regulate splicing. So our lab is interested in answering questions like what drives alternative RNA splicing, how does it affect tumor biology, can splicing help predict risk or survival, is it clinically meaningful, and is it targetable? Well, over the course of today's talk, I'm going to cover 5 different aspects of splicing. And the first one is the differential burden of splicing between normal tissue and cancer tissue, and between cancer of different ancestries, is larger than expected and more complex than expected. So this slide harkens back to the Nature Communications paper. This work originated at the George Washington Cancer Center in collaboration with my colleague, Dr. Norman Lee, and Bidar Wang, who was then a postdoctoral fellow and is now on faculty at the University of Maryland, Eastern Shore. And all I'm showing you here is the principal component analysis of an exon array. And what you can see by looking at the blue circles and the red circles is a dramatic bifurcation between black and white prostate cancer samples as a function of exon arrays. So because this is an exon array, it pointed us almost immediately to this issue of splicing and whether and what the magnitude of splicing differences might be between the two races. So on this slide, you can see that we were actually quite shocked to see the magnitude. So on the left, in prostate cancer, we found more than 2,500 differentially spliced genes as a function of self-identified race, compared to about 1,200 differentially expressed genes as a function of race, with 290 genes overlapping. And interestingly, out of the 2,500 differentially expressed genes that were different by race, 644 of them were also present in the patient's adjacent normal prostate tissue, suggesting that there may be ancestry-related splice variants that are carried forward from normal tissue into tumor tissue and may actually contribute to the biology of the cancer. On the right on this slide just shows another analysis that we've done with lung cancer, in this case squamous cell lung carcinoma. And here you see nearly 5,000 race-related differentially spliced genes. And in both of these, you can see that a large proportion of these splice variants are either generally implicated in cancer or are actually in drivers, oncogenes, or tumor suppressor genes. So these striking results motivated us to expand the analysis to a computational approach to identify the splicing events among tumors that exhibit disparities between Black and white patients. And we did a deep analysis of TCGA splice-seq data sets of all ARS and alternative transcription events for all samples in TCGA. These are the tumor types: breast, cervical, colon, liver, lung adeno, lung squamous, pancreatic, prostate, and uterine. And you can see a varying degree of number of samples from African Americans, pointing out, again, this issue of the low participation of ethnic and racial minorities in clinical trials, in this case, biobanking trials, which is something that's attracting enormous attention worldwide right now. So here what I'm showing is the identification of race-related alternative RNA splicing events and alternative transcription events in TCGA. And the red and blue dots collectively are the top 500 or the top ranked alternative splicing events, the top 500 by race. So those two grouped together. And then below, in just the red alone, are the top alternative race-related alternative RNA splicing events that exhibit an absolute delta PSI of greater than 15% between Black and white patients with an adjusted p-value of less than 0.05. Delta PSI stands for the change in percent spliced in. And that is a computational tool that simply reflects the degree of splicing within any particular gene. So you can see that there are both blue and red dots associated with each of these tumor types. So each of these dots refers to a gene, and you can begin to analyze and quantify the number of genes that show splice variation. And you can do interesting computational analyses, like, in this case, a Venn diagram looking at overlap between breast, lung adeno, lung squamous, colorectal, and prostate cancer. So in this interesting analysis, you can see 18 genes are actually in common between these 5 different tumor types. And you can see an example here of what those genes are. So these are alternatively spliced in a race-related fashion in common to 5 different tumor types. We also were able to look at the top 7, not just 5 tumor types, but 9 cancer types, and the top 7 race-related differences in splicing among all 9 types. And here you can see those genes, and you can see, like, for example, in the red box, ULK3, a dramatic difference in expression of splice variants across all of the tumor types. And then we were able to do pathway analysis, oncogenic pathway analysis. And here, what you can see is all 9 tumor types. And quite interesting and surprising, we were able to find a number of pathways, cancer-related pathways, in common between these tumor types that showed race-related differential splicing. In particularly, the interferon alpha response, interferon gamma response, and inflammatory responses in general. And then also quite interesting, the mTOR signaling pathway showed race-related splice variation in common with multiple tumor types. So just to go a little in a little bit more detail. Sorry, I'm not sure what's happening with this particular slide. It's taking a moment to load. We're also able to show differential race-related splicing events, in this case associated with prostate cancer, as a function of disease-free survival probability. The blue dots show differential splicing with higher PSI in Black versus white patients, where it's associated with poorer disease-free survival. The yellow dots show decreased PSI in white patients associated with poorer disease-free survival. And so you can see SEMA-A6, a well-known prostate cancer-associated gene, XOSC9, as risk factors for African Americans, and then XRCC4 and CYP3A5 are as risk factors for white patients as well. And here I'm just showing that we're able to conduct that analysis across various tumor types, and we won't dig into the details, but if you look in the upper right, you can see some very interesting genes: BCL2, FOX, FAM, SC3H. These are all genes that are well known in association with various cancers, showing differential race-related splicing as a function of different survival probability. Well, one of the questions that frequently comes up when you study splicing is, are the splice variants functional? And here we're simply showing that somewhere between 72 and 82% of the splice variants that we find in TCGA are in frame and therefore are expected to be able to be translated into variant proteins. So in summary, from this computational approach, we identified a large number of race-related splicing differences between Black and white patients. A large number of the race-related splicing differences overlapped across cancer types, and the race-related splicing differences were determined to function in oncogenic pathways and were associated with Black or white cancer patient survival. To go a little bit further, we actually built a new computational tool. It's an interactive and user-friendly web app to enable researchers to mine data such as TCGA in the context of race or ethnicity, and it's called Duke CanSplice. And this is just a screenshot of CanSplice. And here, what you're able to see is you can choose your tumor type up at the top. You can choose whether you want to analyze splicing, aggregate gene expression, correlations between the two, or survival. You can pick your gene name. You can pick which splicing variants you want to look at. In this case, I've selected all. And then you can analyze each splice variant as a function of differential expression as a function of race. You can also move on to aggregate gene expression. And in this case, again, it's a little slow to load. You can input your gene name, and then you can look now, in this example, across different tumor types and compare between the tumor types, and you can look at differential variant expression as a function of race. And then finally, you can look at survival. And in this case, you can choose your gene. You can select exactly which splice variant you want to look at. You can look at a KM plot for differences in survival and an interactive output of the selected splice variant and the gene encoding the selected splice variant as a function of survival. So CanSplice is a user-friendly interactive web tool that allows you to analyze splicing as a function of race and ethnicity, aggregate gene expression, correlation with aggregate expression, and association with survival probability. And here, I'm just showing you the portal that CanSplice can be accessed through. So we were also, as I mentioned to you, in addition to the cis-acting splice elements, splicing factors, trans-acting splicing factors are involved in regulating splicing, and we were able to look at differential expression of splicing factors as a function of race between Black and white patients. Here you can see all 9 tumor types, and then within breast you can see the sub-analysis, basal, HER2, LUM-A, LUM-B versus normal, and in prostate you can see in Gleason score. and we were able to identify a number of splicing factors that show a fold change greater than 1.5 with an adjusted p-value of 0.05 that differ between race as a function of race. And this is an example. The SNRNP70 protein binds to the U1 spliceosomal RNA, forming the U1's SNRNP core element of the spliceosome. And this is just one example of the analysis comparing whites in yellow and blacks in blue. And this is looking at breast cancer and breast cancer subtypes. And you can see very dramatic differences in the expression of this splicing factor as a function of race and in these subtypes of breast cancer. Okay, the second part, ancestry-related ARS drives important features of the hallmark of cancer, as well as drug resistance, and can be both predictive and prognostic. And this is right out of the Nature Communications paper, so I won't dwell on this, but very briefly, if you look in the upper left, you can see in the African-American patients both a long form and a short form frequently showing up in samples from African-American prostate cancer patients, compared to those from European Americans, where the amount, the ratio, and the frequency of the short form is dramatically less, and this was statistically significant. In the lower left, what we did is create siRNA that very specifically targets the splice isoform that's dominant in white patients, which includes exon 20. In prostate cancer from black patients, more frequently, exon 20 is spliced out. But when we use an siRNA to exon 20 and we treat a cell line isolated from white cancer patients, it markedly decreases the migration and invasiveness. But when we knock that long form out, which allows the short form to become the dominant form, migration and invasion actually increases in the cell line derived from African-American patients. And on the other side, in that quadrant, you see that if we design an siRNA to just the short form, it has no impact on the cell line derived from white patients because that form isn't present, but it actually decreases invasion of the cell line derived from a black patient, showing that the short form actually drives tumor, the aggressiveness of this line in culture. We then took the cells in vivo, and it turns out that the short form, exon 20, actually codes for the binding site for a drug called Cal-101 or indelisib. What you can see here is that when we overexpress the long form versus the short form, we drive tumor formation. But in the presence of Cal-101, when the long form is driving tumor formation, it's inhibited by the drug. But when the short form is driving tumor formation, there's no impact of the drug, which makes sense because the drug binding site is no longer present in the messenger RNA. And then finally, you can mine TCGA, and you can do a deep dive on PI3K delta, and you can look at the short form versus the long form. And in this chart, you can see that whenever the short form, the ratio of the short to the long form is longer, is more extensive, survival is lower. All of which together means that the short form, which is the dominant form expressed in African-American prostate cancer, actually drives a more aggressive form of the disease. Well, in this slide, what I'm showing you is that if you go back to exon array analysis and you combine data from prostate, breast, lung, and liver cancer, in this example, you can do all these computational approaches. You can mix and match any tumor type. But in this group, we found it quite interesting that a number of very interesting and important genes show race-related differential splicing in common to these 4 different tumor types. That includes the insulin receptor, CD44, integrins, extracellular matrix proteases, fibronectin, collagen genes. But one particular gene caught one of our postdocs' attention, Muthana Alabo, and he did a deep dive into the LM07 gene, which is a limb domain 7 gene that modulates actin signaling. And in this particular gene, the splicing difference as a function of race is a skip of exon 12. So Muthana did some remarkable work and used CRISPR to very selectively and specifically knock out just exon 12, not knock out the entire gene, just that one exon in the entire gene. And when you knock out exon 12, we find a dramatic increase in migration and invasion in this cell line, in this case, GU145. So just losing this one small exon, exon 12, markedly promotes the aggressiveness of these tumor cells, indicating that this one exon has a suppressive effect on metastatic potential. So we went back to TCGA, which is all primary cancer. And very interestingly, we found no difference in the expression of LM07 between normal and tumor tissue. But then we looked at the Stand Up to Cancer cohort of prostate cancer, which is all metastatic. And there we found a very large difference in the frequency of loss of expression of LM07. So what you can see on the left side of the right chart is the majority of metastatic. The more metastatic the tumor, the more frequent you find loss of LM07. So if you go back in, analyze computationally, you can see on the left, if you just look at high LM07 versus low LM07, whenever the LM07 is low, you have worse outcomes. And on the right, if you do an analysis of LM07 as a function of exon 12, its presence or its absence, whenever exon 12 is absent, you have worse survival outcomes. Okay, the next section is ancestry-related mutations and splice regulatory DNA sequences are more common than expected and functionally important. And here I'm just going to reference two papers that we've published, one in IJC and one in carcinogenesis, both showing that SNPs, single nucleotide polymorphisms, in the splice regulatory regions of stemness-related genes correlate with racial disparities in susceptibility to prostate cancer, and they also correlate with survival in prostate cancer. And these are just some of the genes where those SNPs were detected: TP63, ALD1A1, WNT1, MET1, EGFR, CD44, and GDF15. We also did an analysis of the large cohort of known prostate cancer-related genes, and we found SNPs in splice regulatory regions in KLK3, which happens to be PSA, ATM, mTOR, MPI3K delta, and then we also did the analysis genome-wide, and these are some of the interesting genes where we found race-related SNPs in RNA splice regulatory regions of some very important genes: ATPase, hypoxia-inducible factor, methyltransferase, cytotoxicity triggering receptor 2, PBX, and TREM-L4, all of which are involved in cancer. Okay, the fourth one, alternative RNA splicing and ancestry-related ARS is targetable, both at the level of splice variant proteoforms and splicing itself. And here I'm just going to show one example of some work we've done. And on the left here, you see that our approach is to create splice-switching oligonucleotides. In a very real sense, these are antisense oligonucleotides, but they are targeted very specific to splice junctions and very unique sequences in and around the splice junction. So they're called splice-switching oligonucleotides. And you can use these to interact with the pre-mRNA in a way that you can toggle back and forth between splice variants. And on the right on this slide, I'm just going to show you one example of one that we've done extensive work on. We designed an SSO to the CE3 exon of the androgen receptor, which is involved in the production of a pathological variant of the androgen receptor called ARV7. And the ARV7, when it's expressed, correlates with the development of resistance to hormone therapy. So in this example, we're showing that an SSO that targets the ARV7 markedly decreases ARV7 RNA and protein expression, and it does so. That's shown on the left. In the middle, we're showing that it does that without impacting the expression of the full-length androgen receptor, the physiological androgen receptor. And that's very important because on the far right on this slide, what you can see is we have shown that this SSO decreases proliferation and it restores sensitivity to enzalutamide, hormone therapy. And that would not be possible if all the SSO did is simply knock down the entire androgen receptor and every variant, including the full-length one. So this shows the level of selectivity and the fact that we can actually restore androgen receptor sensitivity to these tumor cells. Okay, and the number 5, ancestry-related ARS influences pathways related to immuno-oncology and may drive neoandrogen production. And I'm just going to show one slide here. We've done a deep dive into immune-related pathways. In this, I'm just showing a couple of examples, one in prostate and one in lung squamous cell carcinoma. And you can see that for both of these, we have discovered a number of race-related, alternatively spliced genes that are involved in adaptive immunity, innate immunity, NF-kappa B signaling, inflammasome pathway, interleukin signaling, and defense path responses to both bacteria and viruses. So we're in the process now of analyzing this at a much deeper level and looking at its biological function as well. So those were the 5 examples that I wanted to show you. And I just want to end with 2 more slides to illustrate how important we think some of these discoveries may be and how important it is for us as a cancer community to really stress the emphasis and the importance of conducting ancestry stratified clinical trials. We do this in the context of splicing, but we certainly strongly encourage this for all cancer-related clinical trials. And just to illustrate how important this is, I'm showing here 4 different trials in prostate cancer. ABI-RACE, which is abiraterone stratified by abiraterone prednisone stratified by race, conducted here at Duke, at the Duke Cancer Institute. Dan George is the PI. The PROCEED trial, which is CYP-T stratified by race. The PI was Oliver Sarter down in Louisiana, but Duke was actually the largest accruer to this trial. Docetaxel study, this was a retrospective meta-analysis conducted by Susan Halaby here at Duke. And then the ongoing Panther trial, which is apalutamide, abiraterone, and prednisone stratified by race, which is in progress, so I can't talk about it. But the first 3, something quite dramatic has happened. In all 3 of these trials, hormone therapy and cellular immunotherapy and chemotherapy, 3 different modalities, 3 different types of analyses. African-American men came into these trials with worse disease by multiple biochemical and clinical indicators, and in each one responded better to therapy, in some cases markedly better to therapy. And I can't think of a better impetus and exhortation for us as translational scientists and physicians to really increasingly stress the importance of conducting oncology clinical trials that are stratified by race and ethnicity. So we'll end by just a cartoon that's in a commentary that we published in Clinical Cancer Research last year. On the left is a tumor cell from one ancestral population. On the right is a tumor cell from another ancestral population. We're just trying to indicate that at the level of DNA, there may be biomarkers as a function of the SNPs that I talked about. with respect to the transacting factors, you can see differences on the left and right, SF1 times 2, and then the different ratio in the other cell on the right. You can see the pre-mRNAs being made and the different splice variants being made as a function of the combination of cis-acting elements and transacting elements. You can see stressing here that neoantigens can be produced by these different splice variants. Biomarkers can also be produced at the proteoform level, and that you can target splicing both with therapeutic small SSOs and therapeutic small molecules, as well as begin to really explore the universe of proteoforms, that is, protein variants coded for by these different splice variants for sensitivity to small molecules. Okay, it takes a lot of people to do this kind of work, and so here I've already acknowledged my laboratory team, but we can't do this work without our clinical partners, both in GU and in lung clinical partners, in MedOnc and SurgOnc, surgical oncology. Here, my key Duke collaborators, our clinical research team who are actually consenting the patients, and of course we thank the patients themselves, our shared resources supported by the Duke Cancer Institute, especially the biospecimen repository team and my outside collaborators. And it also takes a lot of money to do these kinds of this kind of research. So we have multiple grants from the NCI. I mentioned earlier the P20 Patchy grant with NCCU, the P20 Disparities Spore grant, multiple R01s. We have a 2018 Challenge Award from the Prostate Cancer Foundation with Movember Foundation funding. We have 3 DoD grants from the Congressionally Directed Medical Research Program, Department of Defense, both in prostate and lung cancer, and then grants from the V Foundation, the Murdoch Study, Peter Michael Foundation, and several internal grants from the Duke Cancer Institute as well. So thank you for your attention, and I'll stop there.